Okay, this next screencast is on the cell cycle. And so we're getting into mitosis now. And so we're going to discuss what the cell cycle is. We're going to start off with one of my famous animations, which really has everything you need. It's very thorough, but then we're going to break it down into two parts. All right, so let's get the Annie up. Okay, cell cycle animation. There it is. Sorry, I'm being a little bit pronounced there. All right, see you on the other side. See you on the other side.
Most eukaryotic cells follow a process of growth and division called the cell cycle. These events include a growth stage, mitosis, or nuclear division, and cytokinesis, or division of the cytoplasm. Throughout interphase, the cell is engaged in growth and metabolic activities. Interphase can be further broken down into three discrete phases called G1, S, and G2. During the G1, or first growth phase, normal cell functions occur as well as cell growth. In the S phase, DNA replicates, producing two copies of each chromosome. During the G2 phase, the cell continues to prepare for mitosis and cell division. Mitosis, or the M phase, has four stages: prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes become visible and condense, becoming shorter and thicker. Each identical copy of a single chromosome is called a sister chromatid. The nuclear envelope breaks down and spindle fibers form as microtubules grow out of the centrioles that move to opposite poles of the cell. During metaphase, the double-stranded chromosomes line up along the equator of the cell. The spindle is now fully formed, and the microtubules attach to each sister chromatid. Anaphase begins when the sister chromatids of each chromosome begin to separate. The centromere that holds sister chromatids together divides, and the chromosomes move away from each other along its spindle fiber. In telophase, the two groups of chromosomes reach the opposite ends of the cell. As a new nuclear envelope starts to form around each group, the chromosomes uncoil and the spindle disappears. The division of the cytoplasm and organelles is called cytokinesis, or the C phase. The result of mitosis and cytokinesis is the formation of two genetically identical cells. To summarize the entire cycle, G1, S, and G2 phases occur during interphase while the cell is growing and preparing to divide. Mitosis, or the division of the nucleus, consists of four phases: prophase, metaphase, anaphase, and telophase. And in these phases, duplicate copies of the chromosomes are split equally. Finally, the cell cytoplasm divides during cytokinesis and results in the formation of two genetically identical daughter cells.
Okay, that does that. All right, so that's the entire cell cycle. All right. And we're going to break this down. We're not going to do everything that you heard in there. But this is a good animation to come back to because it has everything about all of the phases in there. So let's bring up the PowerPoint for a couple of minutes and we'll run through this quickly. All right. So let's get to the next page. All right. So the cell cycle, again, is the order of events which I guess culminates in the division of a cell into two new cells. So basically the life cycle of a cell. All right. So mitosis is just one portion of the life cycle of the cell. You heard G1, G2, and S phase. So the life cycle and then the dividing portion of it, those portion, and cytokinesis. For here, we're just going to take a quick look at the G1, S, and G2 portions of the cell cycle. So those three phases are what is called interphase. Okay, so basically the period of time between divisions of a cell. Now don't go, you see 10, 9, and 4 hours. I don't know what that's referring to, so don't go with an hour, all right, because some cells don't even divide. So the phase of the cycle is in three distinct phases. So it begins after the splitting of a cell into two new cells. Then that cell has to undergo its own cell cycle. G stands for growth. And you can see inside the portion of the pie what happens during the G1 phase, the growth 1 phase, the first intermediate stage when the cell grows and prepares for DNA replication. So what you see going on there carries out its normal cell processes, photosynthesis or respiration, or both if it's a plant cell, protein synthesis, organelles replicate. All right. The S phase, which is DNA replication, and so all that happens is necessary to make two new cells because each need an exact copy of DNA. So the S phase is the stage where DNA is replicated. The G2 phase is another growth stage. Okay, the cell finishes growing and prepares for mitosis. All right, so, and then the mitosis is broken down into the specifics which you heard. All right, so the cell cycle G1, S, and G2. I'll bring up, I really hate these noises. Why do I take them out? All right. So there you can see what goes on in interphase. And interphase is another name for the G1, S, and G2. All right. We'll discuss what a centriole is again and what it comes, because that comes from way back when. All right. So you take the information off of and specifically know something for G1, S, and G2 in your Cornell. All right. And there you see it there. All right. What happens? All right. Cell doubles, organelles, don't forget also G1, very big metabolic happening. So we leave it there for you. And then this whole PowerPoint will go up as well later on. All right. The cell cycle interphase, that's what this is. And there you can see the S phase, chromosomes replicating. That's what it looks like when DNA replicates, replication portion of DNA.